Hello, everyone, and welcome to the International Carnivorous Plant Society's Roundtable on breeding Venus flytraps. I am Kenny Coogan, the Community Relations and Education Director, and I'm really excited to have our panel of experts on today. I wanted to show off my t-shirt that I'm wearing. It is Utricularia inflata. And if you want this t-shirt, but more importantly, if you want to help support the ICPS, its conservation or education initiatives, you can go to the link below and you can purchase a super cool t-shirt just like this. All right, so this is part one of our Venus flytrap breeding roundtable. And we're excited to have a pretty diverse group of experts. And with that, I will let them introduce themselves before I ask the questions. So Matt, if you want to take it away.
Hi, I'm Matt Miller. Just recently relocated from Southern Oregon to Missouri, Southeastern Missouri. My wife Leah and I run flytrapcare.com, and the associated store is flytrapstore.com.
Hi, Ross. This is Sarah Hunter. We run Hunter Flytraps out of Somerville, South Carolina.
Okay. Hi, everyone. My name is Juan Moscoso. I run Fayetteville Flytraps here in Fayetteville, Arkansas.
Hi, everyone. I'm Stephen Wang, and I'm in Northern California. And we run Evan M. Carnivorium. This is Evan and Emmy. Whoop, there you go.
I'm happy they are able to attend. So we did ask on social media a couple of questions. And first we'll start off with, we're talking about breeding Venus flytraps. What characteristics or what traits do you look at when choosing the parents? And you guys can just have a discussion. So if anybody wants to start.
I guess we can start. So we have a lot of flytraps that we have in our breeding program, and there's a lot of things that we go into choosing parents. But I think first and foremost for us, it is the timing of the flower, and that basically you don't always get, and at least from our standpoint, you don't always get to choose exactly what you want, because the timing of the flowers. Over the years, we've figured out who's are sort of early flowering, who's sort of, generally in the middle, and then who's the late flowering. But you'll see a lot of our crosses, we sell a lot of cultivar cross seeds, and you'll see that our crosses every year, a lot of them are fairly similar, and it's due to the timing of the flowers. So there are a lot of times that you want to choose certain things, but you can't always do that because the flowers aren't timed at the right times.
Have you tried to freeze the pollen? That way you can do breeding with those flowers and on.
That's a really good question. We haven't been doing that. I know a lot of people do that with Nepenthes. We haven't been doing that, and part of it's just that we have so many plants that we're trying to cross, and it ends up being a bit of a mess. And we've got that's part of the reason I have the two kids, and this started out as their project, but they're doing a lot of the hand pollination for us, and so I think that's probably a great idea. We just haven't done it.
To freeze the pollen of mine, because I do have that issue with some of the plants I want to breed flower earlier than the other cultivars, and honestly, I don't really keep record if the freezing works well or not. But I think in some occasions I got seed from those plants that I use frozen pollen. So basically what I do, I just cut the the anther from the base, put them in a little plastic vial that I labeled with the name of the cultivar. And I put that little vial inside another vial with silica sand, and then I put it in the freezer.
Okay, I take a little less, direct approach in terms of which plants I decide to cross. I usually, like Stephen pointed out, some flower real early in the season, like coquillage and cayenne are some of the first to come up. And then, like crimson sawtooth is one of the last, FTS crimson sawtooth. So in some cases, if I want to cross something like that, I'll manipulate their photo period. I've never tried freezing the pollen, but typically what I do is I just have so many plants that I let go to flower. I'll just cross a bunch of random things that I think could turn out something nice, and then sow a bunch of seed and then see what happens. So I don't really track per se direct genetic lineage a lot of the time. Sometimes I'll maybe make 15 or 20 crosses a year where I have, labeled, but most of it I just kind of take a shotgun approach, and whatever's flowering, I cross pollinate and then sow thousands of seed and pick out the best ones.
I definitely identify with what Matt's saying there. I think as far as like choosing like characteristics, one of the things we kind of think of are, I mean, just broad categories. One is trap morphology, one is cilia, and another is coloration. And so we do try to, I mean, for us, trap morphology also includes size, obviously. So sometimes we have a whole system where we're trying to make a larger trap. And then we have another system. Again, this all started with my kids' project, and that was what they wanted, was to try to make a larger trap. And then there's always this interest in long trap morphology. I think we can all agree that Alien and Jaws Smiley and these other cultivars have really piqued a lot of people's interest. So we're that's always been an interest of ours. And as you'll see, a lot of our crosses involve Jaws Smiley and Alien, and that's just our, we have specific goals in mind, and we have been trying to see how much of that is more seed bearer and more pollen bearer. But we've actually been a bit surprised, because a lot of that we felt was more seed bearer. And I think most of us would agree that we tend to see more of the characteristics in the seed bearer. But I've been surprised recently, because we've had a couple of the Jaws Smiley crosses with pollen bearer that we're seeing with longer trap morphology too. So
agree with all of that. We definitely feel that. It's the same thing. I discovered that I've been spending more time trying to breed the plants as of last year. And yeah, I think like Matt said, I just I look up to some of you guys. So I was like, oh, so he has the same problem. Okay, good. Because at the end of the year, it was like, well, I got a bunch of really cool open pollination. But anyway, as far as parents, I totally agree. One of the first things we were trying to do was it was G17 with Polish Dracula, have a giant Dracula, or something actually I wanted to ask, and while I know we're going through these questions and stuff too to discuss with you guys, I know I mentioned it before to Steven, was Schupenstiel, getting the scaling. We've gotten it accidentally. We're looking into, that would be really fun. I'm trying to think of the ones. Actually, I have it written down. I have the sheet around here somewhere. I'll come up with it. But it was like we're Schupen lunatic. Wouldn't that be the coolest thing?
Yeah.
You know what I mean?
That was one we were really trying for.
That's not a good name, I guess. But you know what I mean? We're going for it. What's weird but would look really cool together too. So but yeah, we've had the same issues, it sounds like, as you guys. And actually, I want to try that freezing idea too, because we have had an issue. I think Matt brought it up first about the timing. I actually already have some that are throwing up some flowers early, but you know how it is. Yeah, same thing here, where at the end of the season, things that I wanted were too late. I think my go figure FTS Archangel was the last, so I wasn't able to try to cross it with all the front end stuff, Polish Drac and G17, other things like that. I can't think off the top of my head. But
Ross and I did speak about the Schupenstiel thing. I'm curious if Matt and Juan have had the similar issue. Schupenstiel is actually one of Evan's favorite traps, and we've crossed Schupenstiel as seed bearer, I want to say 7 years in a row now, and we have yet to see any scaly progeny. It's a very weird thing. And then and we've even tried. I mean, Evan actually had this theory that he could feed Schupenstiel. We don't really feed our plants because there's obviously plenty of bugs here, and we get plenty of growth without it. But he babied Schupenstiel for a couple seasons with the hope that we would get somehow better seed. And I mean, we always get good seed, but even the years that we babied it, we didn't get scale on the progeny. Matt and Juan, have you guys seen any of that? Have you guys been able to get shoot and steal to get to reproduce with the same scale?
I can't say that I've done a lot of crosses with shoot and steal, but I haven't exactly tracked them. I can say that I've gotten quite a few seeds with scaling, and I want to say that Trichterfall was the parent plant, and not Schupenstiel. And I think Schupenstiel might be a descendant of Trichterfall. So
interesting.
So if you want scaling, I would say possibly try crossing with Trichterfall. Yeah.
Yeah, I haven't done any crosses with Schupenstiel because I didn't have the cultivar until this year. And the one I have currently is super small. But funny story, so you all know those Flytraps that come in the box that you see at Lowe's and Home Depot and whatnot. I found one with scaling. Out of all those, I found one that had scaling. And I thought it was aphids or some sort of pest, but it was like some mutation that gave it scaling. And it's been consistent for the past 2-3 years. So I'm definitely gonna do crosses with it. It's not as cool as Schupenstiel. It's more like 2 scales in the center of the leaf. But I don't know. It was funny because I always check those plants for mutations. Now and then you find a gem like that. But now I haven't done any crosses with Schupenstiel. Now you guys do a lot of open pollination for breeding. I focus more on more specific breeding. Like if I see a cultivar that's cool but small, I'll grab it and try to get it bigger. So I'm doing like a lot of werewolf with alien, and I hand pollinate my stuff. I label the flowers with the crosses I make, label the seed. I mean, I do everything small scale, but very organized. So I know what I'm getting from the seeds. So I do less. I have less seeds than you guys do, but I do more of like the indeed nerd labeling the flower, polling, and all that stuff to get more precise stuff.
I did that for quite a few years and then was very disappointed with what most of the outcomes were. And I'm like, no, I'm not doing that anymore. I'm just going to do a bunch of crosses and see what comes out of a huge mass of seeds.
I think we're probably where Matt was a while ago. We've been doing this now we've been doing the closing the pollination off with bags and doing all that for the last 5 years. And it's a lot of work. And as our collection has gotten big, I mean, I think we've got like 140 or 150 cultivars now or something like that. And I mean, what I have the advantage of is I got my 2 kids who are, every day after school, they're sitting there doing this stuff in the summer, or it's a huge pain. And I think what Matt's saying is shared in some regard, because I've got all these little trays of, where we've specifically labeled the cross and the seedlings are growing. And 95% of those just look like a regular fly trap. But I will say it's super cool because we've got these little colored, plant labels that are fluorescent, and we go through the trays and we mark the ones that we think have potential. And as each season that goes by, when you see the ones that have potential, you're always like kind of excited that you're going to find something cool. And the number of crosses we do to hit those strikes, obviously it's probably 2 to 3% of what we're growing, but it is cool when you get it. And obviously, Matt, because you've got a ton of them out there, but maybe I could see where at some point you're just like, it's probably better just to do the crosses and grow them out and don't worry about all the labeling, because the labeling just gets to be a mess and it's hard to keep track of all that. But at least for us right now, it's fun because it's what the kids are choosing, and they're, this started as a project. We went into a bit of genetics, and so the kids are trying to choose what traits they're looking for, and it makes it interesting because when you look at the progeny, then you're looking specifically, I picked this cross to make long cilia, or I picked this cross to have an elongated trap. So if I look and we don't see the elongated trap, we're sort of like, yeah, that one's going to the coal zone. But And it's been hard to do that because I'm sure you guys all went through that stage where you don't want to cull anything because you just can't. It's like in your heart, you grew this from a seed and you don't want to cull it. But at some point, sort of your grow space becomes an issue.
It's probably easier to cull the Venus flytraps than show chickens.
Right.
All right. So I want to know, maybe Juan, how do you keep track, just like technically, are you putting labels in the female pots and saying, this is who I crossed, or do you have a notebook, or have you numbered the pots? I think in a webinar, Matt, I referenced you. I saw a video where you had all of your Venus flytraps alphabetical. And in the webinar, I said, oh, I'm going outside after this webinar and I am organizing my flytraps like that. So, and I'm not breeding them. I'm just organizing them so I know where everyone is. So Juan, how do you keep records? And then maybe Stephen, you know, are you putting it on the stem? Are you putting it in the pot?
Okay, so when I hand pollinate a flower, I put a label on that flower, not the whole thing, but just that one flower that I put pollen. I have like little labels that I print out, and then I basically
Like a label maker, like a sticky.
No. I let me see if I can find it real quick. I basically print out in a sheet of paper the names of the cultivars, very tiny, like Alien or DCXL, and I print like DCXL 10 times, Alien 10 times, something 10 times, and then I have a whole sheet with the names of cultivars. Then what I do is I laminate that sheet. Here it is. So something like this with all the names of the cultivars.
Yep.
And it's laminated. So when I pollinate a flower, I cut the name of it and tie it with a string to that flower. And since it's laminated, it's waterproof, so that is, I know what that seed cross is. And then since I'm getting like 50 seeds of the same cross or something like that, sometimes I plant all the seeds in those big 108 like cell like seeding trays.
Yep.
And what I do is I have a system where like you have A, B, C, D, and D, and then 1, 2, 3, 4, 5, 6, 12. So from A1 to A12 is DCXL by Dracula. From B1 to B5 is Alien something. So I write all of that in a notebook, and then I can just, if a lot of seeds died, I mean that's part of the game. And the remaining ones that are that have potential, I know what they are with that system, and then at those I put apart with a label. The other normal looking ones, I'll either sell them or get rid of them, but I keep all the crosses mainly by the A, like blocks and cells, like Excel basically.
Yeah. How many trays for you are you seeding?
I mean, last year I seed about 3 of those.
Okay. So it's manageable.
Yeah. I mean, each cell has like 128. I mean, each tray has 128 cells, but it's not bad.
But it doesn't take up a lot of space that way.
No, I mean, you have 3 cells and with 300 plants, so it's very it saves a lot of space. Now, if I'm planting some other cross that I don't particularly want, but I just want it for selling, I just put like a bunch of seeds in one spot and forget about it. But for the specific crosses I do, I use those 100 cell trays because they save space, and I can easily put them here on my grow lights and just watch them grow.
So I think our approach is similar, although the way we do it is we use colored plant labels, one color for each grow season. So when we do the cross, we cut a standard plant label into thirds, so they're very thin, and then we just write the cross on there, like you know, purple people eater X alien, and we would put that next to the stalk that was pollinated at. If we're isolating and we cover the flower, then that means that every time we pollinate, it's only with that specific pollen. So we don't, even though there may be multiple flowers on a single stalk, the way we do it is each stalk only gets that pollen, because that way, you know, there's obviously, there may be some self-pollination clearly in there, but if it's not self-pollinated, it's going to be from what we put on there. So each stalk is dedicated to that pollen. And, you know, in a mother pot, we may have, you know, 10 stalks. So we put the, you know, the label next to the stalk that got pollinated. And then at the end of the season, you know, that where that is that when we go to harvest, then we, you know, we harvest directly into wax glycine bags or glassine bags. And then we water, so we sell some of our seeds, probably a large chunk of our seeds are sold. There's definitely some that we keep for ourselves, some that don't go for sale, that we were very specific in what the kids wanted. And those are a personal stash. Some of those then we tend to water germinate, like the ones we really want to keep, we water germinate. So we water germinate in test tubes. And then we will take them out of the water and put them into peat. And this each pot is dedicated to that cross. That way we don't mess any of the crosses up as far as that stage. And then, like Juan said, so we grow all of our plants here outside except our seedlings. So our seedlings we bring indoors. We have very limited grow space indoors, but we grow them under lights and we fertilize aggressively for the first season.
And what are you using to fertilize?
So Evan actually, during the pandemic, Evan did an experiment when we were in remote learning. And I mean, basically he chose a bunch of different concentrations of Maxi, and we were trying to look at what point there was an inflection point, or, you know, where we started seeing negative returns. We also he then later did an experiment with Maxi actually for germination. So for water germination, Evan had this thought that, well, maybe we could put Maxi in the test tube and see if that would work. I can tell you that doesn't work because he did that experiment. So putting Maxi in a tube to water germinate seeds is a bad idea. But Maxi as a foliar spray is great.
But it's good that you did that. But it's good that you did that experiment, because now others don't need to replicate that.
And we're going to be hopefully putting a YouTube video out soon. Him and his sister are working on it, but we're going to show you the data from the Maxi experiment for seedlings.
Awesome.
And they did measurements on did you measure every week?
Yeah, I measured every week.
So he was using calipers and measuring traps every week, and we've got data points for all that. So that should be, I think people are going to be a bit surprised at what we found on that.
Very cool. All right, before we go to the next question, I want to stay on this topic. We had a couple of comments. One is from David Zell. Why do people like the absurdly malformed, practically non-functional cultivars? And then somebody else said, and why Sano 310 says, why do people create non-functional mouths in Venus flytraps? And then somebody else said, Saint Pitcher said, I'm especially baffled at the current record holder for the biggest trap. I assume they're talking about alien. What's the use of a big trap if it even does not shut properly? So my Venus flytrap breeders, I feel like some people are hating on the malformed, goofy looking Venus flytraps. What are your thoughts? I don't want to put you say your thoughts and then I'll say mine. I don't want to influence you. So Ross and Sarah, what do you guys, do you favor big traps? Do you favor functional traps? Do you favor the weird?
I like a good B52. We actually have a lot of some of the breeds we've had are like really cool looking, you know, and we do like the reds. Like actually one of our first was Maroon Monster. I mean, hey, it's got that killer lime edge too, you know. But yeah, like I do like collecting very strange traps just because it's fun. It's more like a work of art, I think, than functional. But I have had So like on destruction, I've had them, the side mouths or side heads, I've had them eat, so they're somewhat functional.
But I think it's mainly because you can. You kind of want to see what they can do. Like, how far can you push this plant? Like, I've never seen another plant be able to morph into all these different shapes and sizes before. So even though they're not functional, it's still kind of amazing that they can even do that.
Yeah.
I know this might sound weird, but it kind of reminds me of like dog breeds. They come from a wolf, which is very functional, and you can get a pug. Everybody loves pugs, but it doesn't mean they're the most functional dog. So yeah, it kind of goes to because you can. It looks cool, and it's just kind of fun.
But we like them. I think our opinion is we enjoy them.
Yeah.
We're not going to hate on them.
But I also really love functional traps. Like I love our B52, and we have a cross that we have been calling Tippity Witchet. And I love how giant it is. It's so fast. It grows really well. So I do like functionality as well.
Yeah, and you know what? I did forget that. I do look for division too. And I think we've all noticed some just don't really divide. So and I know Matt is does TC. I haven't touched tissue culture, and I watched his how to, and I was like, I'm gonna let everybody else do that.
Yeah.
I respect it greatly, but anyway, I don't want to take up the whole time here. So
I appreciate it. All right, so Matt, what's your thought on non-functional traps?
Well, I think there's beauty in diversity, and I just love having everything, the whole gamut of fly traps. And if you go through a few seasons of breeding these things, you figure out that it's really, really common to have functional traps, and to get something grown from seed that is abnormal enough to not function is pretty rare, honestly. So it's more of a selecting out the rare varieties because they're unique, and it's fun to grow something that is unique. And then hopefully propagate it, because there's I know it's not the majority and of growers, but a lot of people do really appreciate all the freaky diversity that's makes up the genome of this particular species. That's amazing. The range of plants that you can get, they don't even look like they're the same species.
Within
like
one species. Yeah.
It's all one species. Yeah. And people that come to the hobby, they're like, how many species are there? And I'm like, well, there's just one. But they look very different. If you look at a petite dragon, you would not think that would be related to a DCXL. They look totally different. But yeah, I just think it's an amazingly diverse species. So the more we can get weird genetic anomalies in it, I think it's just cool to see.
All right, maybe we can go to Evan, because I saw Stephen kept looking at Evan. Evan, do you like the non-functional Venus flytraps?
Not really. I like the functional Venus flytraps, because if it's non-functional, it kind of looks like a different species of plants in my mind, and it's not like a Venus flytrap where it can catch flies and bugs and all that.
Yeah.
We also, I think part of it is that we don't, we're really into trying to generate seeds. And I think, a lot of these non-functioning traps just they're so fragile. And I think one of the misconceptions out there, everyone says, don't let your fly trap seed and all this stuff, don't let it flower. I mean, we've been doing it for, 8 or 9 years, and we haven't had any issues, except I will say with the ones that are non-functional and just really kind of fragile cultivars, those are the ones that, if we've had them die, those are the ones that died when they tried to seed. And I for our purposes, we don't. I mean, I don't. You're not really a big fan of those, huh?
No.
Yeah, I'm not either. But Evan, he babies Alien every year to get it to seed. He babies it. He hand feeds it. We don't hand feed anything except Alien and Cheap and Steel, just because we're trying to get those to breed.
Juan, do you enjoy the malformed?
I enjoy them both, honestly. I love Jaw Smiley. It's one of my favorite cultivars of all. It looks like you're growing a piece of mouth on your pot. And so I love those things that look like alien like it's not even from this planet. The weirder the better. I mean, there's some cultivars that are just kind of ugly out there, like fondue or some other ones that are just totally out of shape and they don't look pretty at all. So I'm fine with the non-functional ones as long as they are pretty, you know. Like I've been trying to breed alien with some other ones and make it even more weird looking. But at the same time, I mean, I also breed for functionality. I mean, I want something big that's functional, you know.
Mm-hmm.
Also, I mean, I'm doing this as a business. People seem to pay more for those weird non-functional stuff. I mean, I don't know if you've seen the price of Jaws Miley or some of those. So I mean, in one way, I also do it for the demand. You know.
Absolutely. All right. So speaking of that, how do you select which ones you're going to keep and which ones you're going to throw away or cull? And also, how old are the plants when you select which ones you're going to keep and which ones you're going to cull? So I want to definitely hear from all of you. Juan, you want to start?
I hold them for at least a year or two. So I release I had Stegosaurus, and the first year I had it, I mean, it looked good. I think I left it in cultivation for at least another year to see if it was stable. And so, yeah, I like to see that the plant is stable. And also, I mean, a lot of plants, they look very different when they're in the immature shape. Like Stegosaurus, the first year I had it, didn't have the dentation it had. It was a little bit different. So for a little while, I kept it because it looked different from the others. But after the second year, I was like, oh, wow, this looks much better than it was before. So I like to, if I'm going to release a cultivar, I like to keep it for at least a year or two just to see how it looks mature. Because in my opinion, one of the things I hate or that kind of grind my gears is when you see new cultivars out there released on the ICPS newsletter, and it's just like a little baby plant. You don't need to know what it looks like when it's mature. I mean, is it gonna be stable? Is it gonna look like that? Is it gonna look better or worse? So I think for the consumer side, you know, you want to give them, hey, this is what you're gonna get. This is what it's gonna look like later. So the more you hold it, the better, you know.
All right. So Juan, you had Stegosaurus. You had Stegosaurus had siblings. How many did you narrow it down to? Just one individual plant?
No, on that particular year, that one was the only one that had the dentation. The other ones look like common Flytraps.
Okay.
So I pretty much kept it. But like I said, the first year I had it, it looked a little bit different than the others. But then the second year, it looked like Stegosaurus.
And then we're going to talk about how you register it later, but then you register it. And are you just taking all these leaf pullings, or do you take a leaf pulling and then send it off to tissue culture? Or like you?
I did leaf pullings. The plant produces divisions, like at least 5 or so. It's good. It's a good cultivar for making divisions. So I did the first year I had it, I only had a plan and one little tiny division that I had. And I think from that division, I divided into more. So I kept the original plant growing to see how it would mature. And in the meantime, I had like some other little divisions growing up. And I don't do tissue culture myself. I got a friend of mine here in Arkansas who does tissue culture. So I'm having him do that for me. But yeah, at the beginning, I just had one plant, and then it produced one baby plant. Then I divided that one. So and I got a bunch of like cultivars from last year's breeding that I haven't divided yet, or they haven't produced anything yet. So I'm gonna wait for them to be a little bit bigger before trying to split them up, because they might die, and it's the only one plant I have. So
I do all of my propagation in tissue culture, which is a huge advantage. It's been years since I've started Venus flytraps from seed and soil. So what I do is I initialize them in tissue culture, and I usually do a few thousand a year. And even when they're small, fairly small in tissue culture, you can tell if there's something unusual about them. After looking at so many for so many years, usually you can tell. The only thing you can't really tell is giant, whether it's going to be a giant or not, because some giant fly traps never size up in tissue culture, but you can always tell unique teeth, to some extent growth habit and coloration, and then scaling shows up like prominently in tissue culture. So I've had several dozen plants that have shown scaling from various crosses of Trichterfall and Schuppensteel. So what I do once I see something that's unique enough that's in a jar, I separate it out, put it in a special jar, and then I propagate hundreds of them before I even pot any out. And so by the time I have them hardened off, I usually have several hundred plants that are ready to, grow out and see if it's actually something I want to name. And usually before I decide whether I want to name it, I'll send it to a few of friends to grow for a year or so and say, do you think this plant is special enough? Does it impress you in any way? And that kind of thing. And so that's my process, start to finish. Tissue culture, produce a bunch, get it propagated, send it to a few people, see if it impresses them, and then potentially name it.
When you can first identify, wow, this is extra purple, this is extra cilia, is it the size of your pinky nail? Is it an inch?
It depends on the trait. So a red one, you can tell as soon as it germinates, it's red. Like marginal spine cilia, if they're uniquely shaped like lunatic fringe, you could see it right away. Some of the other traits like scaling, they have to size up a little bit. And sometimes some traits like mirroring, like the mirror fly trap that has the flanges on the side, you won't even see that until you get it in soil. So yeah, it depends on the trait really. But if I see anything unique, I'll just pull it aside, propagate enough to make sure, like Juan's saying, when I started this, I was doing it in soil. And if you got a plant that looks really promising and all you have is this teeny tiny little thing that's less than the size of a penny, you're like, oh my gosh, I hope it doesn't die. So I stopped doing that years ago, and I do everything in tissue culture now.
At it, and I was thinking, like a few seasons, a few years to really see it through. Definitely let it mature, because everybody kind of already said it. Just the way they start off small, and you don't see the traits the same way.
Plus, they might change throughout the seasons.
I'm hoping my thing's not lagging this time, and
perfect. So you started breeding
you guys
last
actually
year?
can hear me, so.
And it stopped.
No, Ross and Sarah, we can hear you.
Okay, no, 2020, and we had a lot less cultivars the first year, so I did stuff. They were separated somewhat, but I think that over time it got a little more obvious what. So jaws seem to like permeate my other from 2020. We have a really cool a couple of jaws. One actually looks, well, I sent pictures. I think all of you, except for Juan, I think you guys have seen some of my pictures. I've sent them to you guys. But anyway, jaws, that seemed to permeate all of them. I don't know what happened that year, but I did notice I had like thousands and thousands of jaws seeds, like jaws by jaws. So, but jaws is actually a really strong cultivar.
Yes. So we've had a lot of jaws crosses like B52, and Maroon Monster, FDS Maroon Monster, and
yeah, exactly.
And we've got some pretty big traps with really cool coloration to it, and we've gotten some interesting teeth looking on some of them as well with the big size. So it's just kind of cool that came from that limited collection we had, but now we have a lot.
but we also something everybody's been talking about this whole time too. You mentioned the culling because we you don't want to let like these things go. And but what we were doing earlier, you guys were mentioning like the process. We identify through numbers for each year, and then so we have X amount of seeds, and each one's a number. Then it's got a letter if it produces divisions. So we can track it, because I think Juan mentioned still used, his other, had his other divisions going, and we've had like our divisions of that plant breed again. So actually, like this year, it's going to be interesting because we have so much stuff.
Kind of overwhelming. Yeah, it's a lot.
Sorry.
All right, Stephen and Evan, and Stephen, if your daughter's there, she's been absorbed by the background.
Yeah, no, sorry, she has soccer practice, so
Okay.
she's gone. Let's see. So Yeah, so we've had several cultivars out now. I think we've got like 5 now. Like Sea Scallop, Tasmanian Devil, Purple People Eater, Skyscraper. There's a 5th one that's going to be published coming up. And it's interesting because I hear what Matt says, and so we partner sometimes with, we partner every year with a couple tissue culture folks as well. And the early cultivars are all from them in our partnership. The last 2 and our now current group of plants that we're looking at now are all our own seed grown. And so you can see that the lag is almost 3 years. So Matt's got a 3-year advantage by doing tissue culture because he's got, you know, he can get stuff out in 3 years. You can, for sure, in tissue culture, you're looking at an adult plant and you can get all the characteristics out. These latest cultivars for us are, you know, seed growns from a long time ago. So, you know, we've been doing this seriously now for about 7 years. So, you know, we're now just getting to the ones that are our own seed grown cultivars. We don't, you know, and Matt made some very good points. I will say this. I think trying to come up with a giant by tissue culture is very, very difficult. And I think one of the things I'm unclear about, but I've purchased tissue culture plants, and I will say even giant cultivars that are tissue cultured will divide for I don't know how long based on, and that's where I'm not sure if there's some sort of genetic imprinting by being in tissue culture, or if it's just residual from the hormones or whatever that's in the tissue culture. I'm unsure of. But my feeling is that the true giants that you're looking for are going to be better grown by conventional means. So, and that to me so if I'm looking for a giant cross, and this is where kind of knowing what you're crossing and what you're hoping to get out of it, when we do giant crosses, we don't send them to tissue culture. Those are all done, but we all grow those in-house, and we grow them by seed, and we don't tend to cull those for a long time. Actually, to be honest with you, most of those we still haven't culled. I mean, I've got like trays of these from the last 5 or 6 years that we're still trying to pick out which one. They're all huge, but I'm trying to pick out which ones are bigger than the others. The other things though, like if I know I'm looking for long cilia and I go through the tray and I don't see the long cilia, you know, we tend to let those go early because we have limited grow sprays. And, you know, the limiting factor is my wife, who's like, get that off the patio. You know, I want my table back. Right. So that's kind of what we've been doing. So, you know, if it's a long trap morphology, I've noticed that takes a little bit longer. And Matt knows in tissue culture, that's easier to see way earlier. So some of those we ship to our, you know, our partners in tissue culture, but we always, our prime crosses, we always keep some ourselves, and we still grow those out ourselves in case, you know, it's a random event. So,
just to follow up on what Steven said, it is hard. It's not, I wouldn't say you can't find a giant cultivar by doing it in tissue culture. The thing is, it's just, you can't tell it going in, like before you put them in soil, how big it's going to get. And then even after you get it in soil, it may take 2 to 3 years, depending on how much you want to baby each individual plant before you know. And sometimes you'll have giant fly traps that just will not size up for a few years for whatever reason. Like I'll have a pot of some giant fly traps and they just keep dividing, you know, something happens, sets them back, whatever. So just culling for giant fly traps is really hard. And then in terms of plants dividing coming out of tissue culture, kind of really depends on what the person on the other end of the tissue culture is doing. So I don't use any hormones at all for my tissue culturing of fly traps, 0 hormones. So when they come out, they don't really have any kind of residual effects. I do give them some certain types of things to make them grow larger in tissue culture that I figured out over the years. But you know, I've noticed a lot of novice tissue culture people will produce huge clumps of really teeny tiny plants and then pot them right out. And you can avoid that if you know what you're doing.
So Matt, your plants then tend to, I mean, I don't do tissue culture. I'm just curious though. So when they come out of tissue culture, they don't have that drive to divide then? Because I've gotten plants from some tissue culture folks where you feel like they're just constantly wanting to divide. And even if they're sort of larger you know, and I know that I have a certain, you know, plant that's been, you know, one of our in our mother plots who doesn't divide very much, but I've seen some tissue culture of the same cultivar just like for 2 or 3 seasons constantly divide and not wanna size up. You know, I don't know if that's due to tissue culture, but that was sort of my hypothesis.
Yeah, it very well could be. I mean, if somebody used way too much of a cytokinin of some sort, it would just sometimes if you mess up and you put the wrong chemical in a or too much of the wrong hormone, it can take years for it to grow out of you know, if
Okay.
you really hopped them up too much. That is certainly possible, but if you know what you're doing, and like I said, I don't even use hormones at all for my fly trap cultures at this point. So um, typically, you know, I go through phases to where they're not dividing much typically, but there are certain cultivars, for instance, Wally, Big Mouth, Scarlatine. There are a few that just never size up at all in tissue culture, and they will tend to divide regardless once they're out, even without any hormones or anything. So.
Yeah, so I think I have 2 more questions, and it's about tissue culture, or maybe it's about cultivars. I know a past webinar that we had Damon Collinsworth on. He was, and Daniela, so I think it was just like a Q&A. A lot of people were asking or commenting that certain traits that they bought the Venus Flytrap specifically for don't reveal themselves in the springtime, or you have to wait until the, you know, the end of the summer, or they don't show until the fall. So is that related to the cultivar? Is it related to tissue culture? And then can you have a cultivar and then 3 years later that turns into a typical, not, you know, can it lose all of its characteristics that made it the cultivar? So Matt, you want to take the first swing?
Absolutely. There are quite a few out there in circulation, and part of the reason I stopped propagating some of these that became popular was like so Biohazard and Cheerleader. After a few growing seasons, they will often not produce any kind of unusual traps. A lot of the pom-pom traps, there's quite a few varieties now, especially from Green Jaws in Germany. They will not produce palms consistently, and often it's very seasonal. Like I imported a bunch from Matthias Meyer from Green Jaws last winter, and all summer the majority of them did not produce any pom-pom traps, and now they're starting to produce some as they're starting to break dormancy. So that trait in particular, and also the biohazard trait where the fringy on the side of the traps, both Biohazard and Biohazard 2 that I've grown are similar. They'll revert to looking typical often, and for years sometimes. Like I think Biohazard, I had it for years, it didn't do any kind of fuzzy traps on it. So yes, definitely seasonal, and sometimes just they don't produce the traps for whatever reason for multiple seasons.
Stephen and Evan, that would be a good project for you. You could take the reverted traps and try to breed them and see if their recessive traits boil up back up or something.
Good.
Okay.
All right, send us the plants. We'll do it.
I personally am not at the level of you, but maybe somebody else on the panel can do that.
So yeah, I've been hearing that for a while, and we haven't had any do that yet. I've some of our more, you know, our Biohazard 2, our Destruction and stuff, we've only had a couple of years, but we haven't seen any of that. Now, what I have seen at the beginning of, for example, this fall, when the summer traps were dying off and the fall traps were coming in, on our biggest Destruction, for example, most of the traps that came up, we're in South Carolina too, so like it doesn't really get cold till what, November or something, I don't know. So anyway, we've seen on those cultivars, we haven't seen them really fully revert, but I have seen them briefly, like you were saying, for the season, like maybe part of spring, part of fall. For example, jaws, I think it even says in the cultivar description that the triangular, shapes don't really present until summer, which is true,
And
but I haven't seen anything revert fully for years.
yeah, not for years, but we also have like mirror, and that won't make the mirror traps or anything like that for part of the season, and then it seems
it'll
to
just
come back.
divide like crazy.
Yeah, and so yeah, we've noticed a lot of seasonal, but not so much for years. Little bushes of mirror, they just keep turning into all these little fly traps and these bushes, and I have all these pots of all these little bushes.
We have a lot.
They just keep dividing but not getting any like girth to them. They're not doing a whole lot in that respect. So I don't know, maybe a couple more years they'll turn into a real plant.
The other thing that, Kenny, that we've noticed is some of the giant cultivars. I mean, I think like DCXL pumps out some really big traps early in the season, and then kind of fizzles a bit. But that's one of the things we've been hoping to develop is a giant cultivar that's sort of giant uniformly all year round. And like, for instance, for us at least, like BCP Z11 is, it doesn't have the largest traps, but they're pretty darn large. And for us, it's all the traps on the plant and pretty much uniformly the whole season. So, as far as giants go, and that's sort of still been our, we're doing a lot of breeding stuff, but our goal, much of our goal is trying to develop a giant cultivar.
Yeah, that's always kind of the goal for us too. I don't know, just having a giant trap just seems to be pretty fun.
It's at least what the customers or the consumers imagine when they think, if they're new to carnivorous plants, like, oh, Venus flytraps are huge.
How big did Tiffany get? Was it about 2 inches? Yeah.
Good. Send a note to the Guinness World Record people.
We'll see you, Jeremiah.
All right. So the last question is, could you name maybe just like one or two growers that you are admiring their work currently, or cultivars that you really like? So Ross and Sarah, you want to start?
Yeah. Hey guys, how's it going?
Yeah, you're right there.
No, yeah. Yeah, I already am a big collector of FTS's cultivars, and actually I know Stephen, you worked with Craig a bit too. The only thing I didn't get Red Incredible Hulk yet, so I need to get that.
Yeah.
But yeah, I would say as far as breeding, actually, there's just so much good genetic material just between the two groups there. So really, we did this past season. We used quite a few FTS and Craig's and Stevens. So
well, I know some of my favorites are like Lunatic Fringe and Maroon Monster. They're huge. They're huge, and they're just so interesting looking. I don't know. I always have to grow those.
Yeah. Quick mention. Yeah. I don't know. I think I shared it with you guys. I don't think Juan, but those weird. I still wish I could have gotten more scientific results for everyone. But those little nubs fringe I had last year, they looked like another plant growing off of a trigger hair. That was like the strangest thing, and it did it for like 2 or 3 traps. And then they died off just like, as the trap died. And
it was pretty cool.
No idea. I mean, I'm in track it this year, but that was probably the coolest thing I've seen so far. Very alien.
Yeah.
Or,
You know if that came from tissue culture or seed grown?
I bought that one directly from Matt and Leah, so from FTS. So I'm guessing yes.
All right, Matt, do you want to say a grower or cultivar or two that you really admire?
Yeah, it's been hard to keep up with so many releases coming out the last few years, but yeah, Stephen and Craig have done great with a ton of different plants coming out, and then I've Ross and Sarah here too. I've seen a few photos recently that are jaw-dropping plants. They look great. From Europe, obviously Matthias, a longtime player, has put out a lot of plants over the years, but I really like the Carnivoria, David Svark. His plants, if you like freaky ones, he has got an amazing array of selection of really unusual plants. So right now he's probably at the top of my list as most underrated. But yeah, those would be my mentions off the top of my head. I have so many plants that I love. So if I went through my collection, I could say, oh, that person, but yeah.
All right, very good. Everyone stay tuned for part 2 of Venus flytrap breeding to hear some comments from David. All right, Stephen and Evan, do you have a grower or cultivar that's your favorite or that you admire or aspire to rather?
I'll let Evan start. What do you want to say?
So we kind of like a grower that we admire is like Drew Bowen and Craig.
And I'll say Matt is the godfather. I mean, that we've got obviously a lot of our cultivars, you know, parentage goes back to FTS. So, you know, he's the godfather of a lot of the stuff we've been doing. And you know, Ross is, like Matt said, Ross has had some really jaw-dropping cultivars come out, and Juan too. So but I will say the guys I really admire right now, you know, just I don't know them like Matt does, but I've seen GJ's stuff and I want to get my hands on it. It's a little pricey for me right now. We're still, you know, having to pay rent in Northern California, so I'm waiting for Matt to drop me some plant love from GJ.
Definitely the guy from Green Jaws. I forgot his name. I mean, he has some very gnarly looking fly traps out there that I mean, those are great. I mean, I don't know how long he took for that, but I aspire to have some crazy gnarly stuff like the way he has. And any particular cultivar, I mean, so far I'm really into the new wine mouth. I'm sure a lot of you've seen it, but it's such a crazy shade of red. I mean, I've seen a lot of new cultivars. I always keep an eye on everything that's new, and I feel the more cultivars come out, the harder it is to come out with something actually unique. And that coloration of wine mouth is just gold for breeding. I mean, you can just wait to get a hold of some of that and breed it with the stuff I have and just intensify it or make it even more weird than it is.
Wonderful. All right. Well, I want to thank the panel for joining us today. And I hope everybody learned something about breeding and selecting baby Venus flytraps.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.